Over the past few years, the pharmaceutical industry has seen an increase in novel biotherapeutics and biosimilar drugs. To support the research and development of these biopharmaceuticals, researchers have been looking for better and more efficient ways to perform antibody quantitation in clinical studies. The ELISA method, paired with UV spectroscopy, is often used to detect and quantify biotherapeutic drugs such as antibodies. Some labs are also starting to take advantage of the accuracy and sensitivity of LC-MS instruments. But traditionally, the sample prep has been time-consuming and includes the troublesome steps of capturing, denaturing, reduction and alkylation, and tryptic digestion. Both of these methods are widely accepted but far from perfect. That's why Shimadzu developed the nSMOL Antibody BA Kit. nSMOL stands for nano-surface and molecular orientation limited proteolysis. It's a proprietary, groundbreaking process that combines nanotechnology and LC-MS analysis to dramatically improve the speed and accuracy of monoclonal antibody bioanalysis. It's a ready-to-use reagent kit optimized for capturing antibodies from blood or other biological samples using an immunoglobulin collection resin. nSMOL then enables selective proteolysis of the Fab region of these antibodies using trypsin-immobilized nanoparticles. The collection of Fab-derived peptides are easily quantified via MRM measurement on a high-performance triple-quadrupole liquid chromatography mass spectrometer like the Shimadzu LC-MS 8060. Here's how it works. First, the test sample is mixed with nSMOL immunoglobulin collection resin and a binding agent. The collection resin is covered with nanowells. At the bottom of each nanowell is a protein A ligand coating to capture antibodies. During 2 wash cycles, the protein A ligands capture the antibodies and hold each one in place and in the proper orientation so that the Fab protein region is facing out. Non-antibody material or matrix is removed during the wash cycles. Next, a reaction solution containing trypsin-immobilized nanoparticles is added. The nanoparticles are perfectly sized so that only a small part fits into the nanowells on the collection resin. In this way, only the Fab region of the antibody is exposed to the trypsin. This selective digestion of Fab peptides decreases sample complexity and limits contamination from excessive proteolysis. It's one of the things that makes nSMOL truly unique and effective. No capture antibodies or ligands are required, and there is no need for solid phase extraction after reaction. Once the sample is centrifuged, the Fab peptides can be simply extracted and injected directly into the LC-MS. The analysis takes only 5 minutes. The nSMOL Antibody BA Kit provides prepared reagents and protocols applicable to a wide variety of biopharmaceutical antibodies. nSMOL leaves other methods far behind, dramatically improving response and quantitative repeatability. It's the faster, more efficient, less expensive method biopharma researchers have been waiting for. For more information on how nSMOL can help you improve your antibody bioanalysis, visit www.labtotalbio.com.
Excellence in science. Shimadzu.